Thank you so much. Good to be back. And Zella would just ask me about my wife. She's not with me this time, but it's not because she didn't like her trip. It was just that we didn't have a task for her that we felt warranted the expense of bringing her over here. But anyway, so yeah, my topic this morning is epigenetics, and can lifestyle really turn on good genes and turn off bad genes? You know, let me just jump right into it. I had the privilege of working in the Marshall Islands doing research on diabetes. You know that right after World War II, in 1953, there were only 3 people in all of the Marshall Islands that had type 2 diabetes, and today it's getting close to 40% of the people that have diabetes. And so the U.S. government spent about $2 million on a research project over 3 years to use lifestyle to change the course of that epidemic, and it was marvelously successful. You can see our treatment approach was lifestyle change. And I'll be honest with you, most of the people that were there on the island, and especially the medical community, thought it was a harebrained, crazy idea, really. And I knew that we had to prove, you know, our proposition. In other words, we were not given any quarter at all. And but here's why it was so important to them, because type 2 diabetes, which is often called adult diabetes, compared to the ordinary risk of heart attack and stroke, if this is the average non-diabetic's risk, which, by the way, heart disease kills almost half of Americans, okay? But if you have diabetes that starts after age 45, it triples or almost quadruples your risk of death from heart disease or stroke. But if it starts younger, under 45, look at the risk numbers there, from 14 to 30 times the risk. So it's obvious to the Marshallese the ministry's department, that this is going to absolutely consume them, because these people that with the type 2 diabetes are definitely going to be suffering heart disease and stroke. By the way, to put this in perspective, do you realize the heart disease is the thing that we, in the U.S., again, I don't have Australian figures, but in the U.S., we spend more of our healthcare dollars on heart disease than anything else. So heart disease is very expensive to treat. You know, you can have bypasses and transplants and all kinds of very expensive. And to put it in perspective, if a hospital in the U.S. does transplants and bypass surgeries, if they're a hospital that can do that, that is their most profitable line of business that they do. So that helps in economic terms put it in perspective. It is a very expensive disease to treat, and it's a very profitable operation to do. Kind of goes together. So here was our results of the first group. I just chose the first 10 days from our first treatment group. Now, if you know anything about blood sugars, these are, by the way, in American values, milligrams per deciliter, not millimoles. My apologies. I didn't think about my audience. I should have converted this slide. But anyway, the point is that this is way too high. They should be running somewhere under 100. And so you can see they started off way high. But after 10 days of treatment, this is actually 2 weeks, but 10 days of treatment, the number, well, it had dropped. This is a regression, what we call a regression equation. They had dropped 74 milligrams per deciliter in 10 days of treatment. And the people were, the patients themselves were absolutely astounded that this could happen to them, because they think of diabetes as a death sentence almost when they get it. And so what did we do? This was the result of feeding them 3 meals a day, 5 days a week, and walking with them and teaching them to exercise about 45 minutes a day. That was, and we had to take some of them, their medicines they were overdosed on their insulin. We had to cut their medications as they were, sugars came down. But anytime I show that, here, now let me get to the real topic. And that is, people always say, well, you know, that's wonderful, but I just have bad genes. You know, my grandparents had this and my parents, and so I've got it. Do you realize that we don't only inherit genes, we inherit lifestyle? I don't know how many of you ladies, during your early years of your marriage, decided that maybe you better learn how to fix something that your husband had really enjoyed his mother made. But oftentimes that happens, that we men want to this dish that we grew up eating. And so my point is, just to illustrate, that we inherit lifestyle, and it's not just genes. Now, in the Time magazine, this is the January 2000, I believe, 2010, January 2010 issue was about, whole thing was on why your DNA isn't your destiny. And it was talking about the new science of epigenetics. And it reveals that the choices that we make change our genes. Now you're like, well, how can that possibly be? I thought genes were fixed. But I'm going to show you how it does change our genes, not the DNA, but it changes our genes. And those of our kids, that's the most amazing thing, especially for any of the younger ones in the audience that are still, what we say medically, of fertile age. But if you're still able to procreate, that is especially powerful, that it can affect your kids. Let me show you that. Well, the story really begins, I've got to move fast now, but in 1950s, Watson and Crick received the Nobel Prize for their discovery of the DNA, deoxyribonucleic acid, was the molecule of inheritance. And then about a decade ago, Dr. Francis Collins, who's now the director of the National Institutes of Health in the U.S., he and his team sequenced the human genome. And this was about 3 billion base pairs. So that DNA molecule is, do you realize there's almost 10 feet of it, almost 3 meters in each cell in your body? It's remarkable. It's very long, about 30,000 genes in there. Lots of variations. These variations, some of them actually cause disease, like sickle cell disease. It's just one mistake. Can you believe that? One mistake out of the 3 billion causes sickle cell disease. But the vast majority don't cause any problem at all. They're just silent differences. And that's how our DNA testing tells you apart. If we did DNA testing here of everyone in the audience, what we would be doing is looking for these individual variations between us. It's better than a fingerprint. Now, this picture here is to remind me to share this with you. You know how, just think back to your high school biology. We all came about from two cells, one from mom and one from dad, right? And did you know that those cells were incomplete? They each had half of the set of chromosomes. And when they came together, we call that fertilization. Now you have a cell that has a complete set of chromosomes, 23 chromosomes. And that cell lodges, hopefully, in mom's uterus and began to grow. And it doubled and tripled and quadrupled. And pretty soon we had trillions of cells. And 9 months after this fertilization, we were born. So every cell in your body has a copy of that first set of chromosomes, right? You know that, if you think about it. And so every cell in our body has the same 30,000 genes. Now, I have a question. If all the cells in our body have a copy of the same DNA, the same genes, how does it make such different things? How does some of it make an ear, and some of it a toe, and some of it makes brain? Do you realize that all the genes for my liver are in my ear? Aren't you glad that they're turned off? I am. In fact, all the genes for a right ear are in my left ear. And I'm glad those are turned off too. So what we've discovered is that the majority of the genes in every cell are turned off. And in fact, it's the concert of turning genes on and off during development that make us what we are, make us this unique organism. Okay, now, so that insight has really changed our understanding about lifestyle and genes. Because you see, we don't actually inherit DNA. We often think of it that way, but what we inherit from our parents are chromosomes, and I already mentioned that, right? Those are only about 50% DNA, and the other 50% is protein molecules. And these protein molecules carry important information because each gene has to have some way to be turned on and off at the right time, right? A switch, like the lights, you know. If these lights had no switch on them, they would actually become a bit of a problem. So we had to be able to turn them off when we want to be able to see other things. So this is a little picture of the nucleus, and here's all this chromatin and chromosomes that are sort of unraveled and ready to be used. And it takes a lot of machinery inside that cell to turn these switches on and off. So the protein molecules provide the switches. And the most powerful way to change the switch setting is something so simple it's almost impossible to believe. But it's eating. Isn't that wonderful? I mean, I love eating. I don't know about you. And a few things I really enjoy more than some good, tasty, healthy food when I'm hungry. But it turns out that science in all of our work so far, and I say their work, I mean, I'm not actively engaged in this. I have done my research on reading about it, but I don't do this kind of research. I do lifestyle change. But nutrition changes more switch settings than anything else we've discovered. And so there's a whole field called nutrigenomics that's grown up trying to discover which specific nutrients turn on which genes and so forth. It's turning out to be a whole lot more complicated than we ever imagined because it seems that the nutrients turn on and off a lot of genes, not just one. You know what I mean? It's like a concert, not like individual. Well, anyway, so let me tell you about this little story here. I got three little experiments I want to tell you, and then I'll close. This one is about a mouse, the Goody mouse. And the Goody mouse is a very expensive mouse because it was specially genetically modified so that it has the genes that cause obesity, heart disease, diabetes, all these diseases that are afflicting mankind, so they can be studied in a mouse model. Okay? And you can understand, I assume, easily why that would be nice. One thing is that their generations occur much faster than human beings. And also, they're more compliant. If you want to do something to them, they'll kind of go along with it. Humans aren't always like that. And so they did an experiment. These mice are usually used to study drugs. But some researchers at Duke University about a decade or more ago got the idea of let's see what we can do with lifestyle change to affect those genes. And they were specifically interested in, could we do something to the genes that have been engineered that would affect future generations. And so they fed this mother a special diet that was designed to turn on and off genes while she was pregnant. And then they wanted to study what happened in the baby. When the baby was born to this mother that had been fed this special diet, really history happened. And it is still the ripples are still being found. You can find this story on Discovery channel where they talk about this. But the mouse here, you see, the baby didn't even look like the mother. Didn't have that distinctive color of coat that they had engineered to easily be able to identify these genes. In fact, they had to do DNA testing, and DNA testing revealed that this mouse has the same genes for disease that the mother has. But they were silenced. They were turned off. They were turned off in utero. So the mother's diet changed the switch settings in the baby before it was born and had results after birth that we could easily see. In fact, the researchers say that the effects were evident for 3 to 4 generations. And that's their choice of wording, but as a Bible student, I couldn't help but hear those words were amazing to me. That's right out of the Exodus 20, right out of the 10 Commandments. 3 to 4 generations. But you know what? Did you realize that ovum, that egg that came from mom that you and I were formed from, that egg was formed in her ovaries while she was in grandma's uterus. So sometimes we forget the connectedness that we have of generations, because the ovaries are developed in the first trimester or so of development. And then I want to tell you about this little rat study. See, that first study that we looked at in the mice, they fed a special diet to turn on and off genes. Now here, in this study, they're just simply feeding this pregnant rat a high-fat diet. This is a diet that you could easily get at any fast food place. You could cook it up at home if you wanted to. Hopefully you don't do that. But it was about a 40 to 50% fat diet. And they fed this diet to the mother while she was pregnant. And they were able, with some very elegant research, my undergraduate's in biochemistry and in molecular biology, so I really appreciate how elegant this research was. They were able to identify which chromosome in the developing fetus was being overexpressed and which gene. And they found that her high-fat diet that she was eating was causing this gene to overexpress, and it was producing anatomical changes in the brain of the developing baby. There were more neurons that were being produced than would normally have been. And these neurons, this is neurogenesis, they took residence in the base of the brain, and they were hormonally active. So this mother's diet was producing an increase in hormonally active cells in the brain of the developing baby. And after birth, during adolescence, these hormones produced, extra hormones, were now causing this baby to have high blood lipids. That means cholesterol and triglycerides. So they were having high, and they had more food intake. They preferred fat. If you gave them a choice, they would choose fat diet over a regular. Early onset of puberty, higher body weight. Do you recognize that profile? I don't know if you have it here in Australia. I do know, and you do. We have it in the U.S. really bad. This would be a profile that would fit the majority of what's happening, and why pediatricians in the United States have recently been recommended to start testing cholesterol levels in children as early as age 2, and to start treating them with statins by age 8. I wonder how long we can afford to do that. I don't think we can afford it now. And then I want to end with this human study, because I've talked about mice and rats, but we're really humans. This is amazing. This is amazing. Well, sometimes we seem like there's some question about it. But this study was published in 2008 by Dr. Ornish, and I'm sure you've heard of. And it was an amazing study. It was 30 men with prostate cancer. So this was a study looking at men that had prostate cancer, and you may know that prostate cancer is one of the more prominent cancers in men today. And if you're a smoker, you might be more likely to have lung cancer, but if you're not a smoker, more likely to have prostate cancer. So they didn't have surgery or radiation. They took a lifestyle treatment program. And this program, they also donated prostate needle biopsies. So they literally took biopsies of the prostate cancer. So with the needle, we can actually take some tissue that's cancerous. And they did this at baseline and at 3 months. So this was two different specimens. And from that, we could purify what's called messenger RNA. This is a molecule that's created in the nucleus when we transcribe a gene, and then it becomes a way to know which genes are being active and which ones are not, because the messenger RNA only has a half-life of a few hours to a day or so. And this is a little description. I'm not going to go into it, but they had a pretty normal diet that any one of us could eat. In fact, I suspect that many of us in this Adventist church are eating this kind of a diet, actually. But now I want to show you what happened when they looked at the results. And this is what we call a heat map. And you interpret it by just looking at the colors. And you just look at this side of the map before the intervention and this one. And you can just see that in general, there's a lot more darker red, colors over here than here. Because if you were to count this, there's 30 columns on each side, for one column for each man in the study. And then there's each row is a different gene that we were looking at to see the level of messenger RNA. Messenger RNA that was present, the darker the color is put in the little square. So it becomes a way to, in a glance, to see that we definitely had more genes going here than over here, because you can see a lot more green and light colors and so on. And what we're talking about here is this is the ontology group of genes that are known to be associated with cancer growth and production. And you can see what happened was they went down. Many of them were turned down or off in 30 days on this lifestyle intervention in males, humans. So this is my proof to you. I'm using this study to show to you that this principles that we're talking about of lifestyle turning on and off genes works in human beings. And now we still got a lot more to learn. Dr. Jerdel is the gentleman who did the study in the agouti mouse that started this, sort of, you might say. And listen to what he has to say. Epigenetics is proving we have some responsibility for the integrity of our genome. Before the epigenetics, genes predetermined outcomes, and we still have people who are thinking that way. But now everything we do, everything we eat or smoke, he says, can affect our gene expression and that of future generations. Epigenetics introduces the concept of what? Of free will into our understanding, our idea of genetics. This is so important, I believe. This is why I always try to share this with my patients in my lifestyle medicine practice, is because I don't want people to get the idea that they are powerless, that they're a victim of their inheritance. It's not so. It's not so. In fact, Dr. Feinberg from the Johns Hopkins in United States, he wrote in the Journal of the American Medical Association that same year, 2008, that epigenetics is at the epicenter of modern medicine. This helps explain the relationship between an individual's genetic background, the environment, aging, and disease. And so this concept of epigenetics is going to permeate all of our understanding of lifestyle medicine and of genetics. So let me just summarize this with 3 things here. You change your diet and your lifestyle. For example, exercise also turns genes on and off. A recent study just published showed that the genes start changing in 20 minutes after you begin within 20 minutes of exercise, beginning exercise, genes are turning on and off in your body as a result of that activity. So I can't wait till we find out how long it takes for food to do it. We don't know how soon it happens. You change your diet and lifestyle, it changes your epigenome. I've just sort of demonstrated that with these studies. And then that changes your gene expression. And if you change gene expression, you can see that literally changes you. That will change your body to be different. And so this is powerful. I love it. To me, it's exciting. And it puts it to a large extent under our control. Now, not everything. You cannot change the DNA sequence, but you can change whether that gene is turned on or off to a large extent. So if you summarize it, if you think of health as the phenotype, we find out that DNA sequence has about 10% of the effect determines our health. Not nearly what some people think. Medical care may be another 10%. Sorry, we doctors have to live with that. It's not that big of an influence. And the environment may be about 10%. But lifestyle, which is largely under our choice, is about 70%. So this is encouraging, I think. Well, this afternoon, I know I'm going to say something about this later, but I just wanted to tell you that I'm going to have a talk that will really tie this together some, and it's about 2 hearts in the human body. Did you know you had 2 hearts in your human body? I want to ask you, do you really want to have a transplant? So you're going to come and hear this. I think you'll find it informative and very, very interesting. Thank you for your attention.